Microscopy is simply the study and use of microscopes to observe objects and structures that are too small to be seen with your naked eye. Microscopy, it's essential, it's required. I mean, of course, we use it in biology. It's also used in medicine, materials science, nutrition, other fields. Microscopes magnify tiny little structures, and they enable the detailed study of cells, tissues, microorganisms, and even individual molecules when you get down to electron microscopes. Light microscopes. They use visible light to illuminate spectrums. They're usually limited to about 1,000x magnification, and they're really good for viewing live cells, tissues, and microorganisms. That's one of the types of microscopes that we easily use in in-person labs. Electron microscopes use a beam of electrons for imaging, and they achieve much higher magnification and also resolution or precision than light microscopes. We do not use them here at FLCC. We do not have one of those. A transmission electron microscope can render a detailed 2D images of like internal structures. A scanning electron microscope gives you 3D images of of surface structures. So you can't have your cake and eat it too. You're either going to see the inside of a cell in 2D or the outside of a cell in 3D. There are also confocal microscopes that use lasers and fluorescence to produce high-resolution 3D images of specimens. And this is really common in neurobiology or the study of your nervous system. Also, using lasers and fluorescence to produce those images is a true form of artwork. Once you see some of those types of pictures, they're fascinatingly beautiful. Cell theory is one boring topic. No, I'm kidding. It's not actually. It's just very simple, and it's one of the foundational principles of biology, and it explains the connection between structure and function of all living cells. It was developed in the 19th century by a couple people, just a few that took part: Matthias Schleiden, Theodor Schwann, who's known for myelin Schwann coverings on cells, and Rudolf Virchow. It consists of three main tenets. First, all living cells are composed of one or more cells, and this makes cells the basic building blocks of life. So viruses are not living, they're not cells. Second, the cell is the fundamental unit of structure and function in all living things. And what this means is that all processes that occur within cells are through their interactions. Third, all cells, this is my favorite, I'm sorry, all cells arise from pre-existing cells. I'm going to let that sink in for a moment. Okay, moving on. And this highlights the importance of cell division in the continuity of life. So, I mean, you should be asking then what made the first cell? Anyway, that's another class. These principles established a unifying framework for understanding the nature of organisms, whether they're unicellular, like the bacteria you see here on the left, or whether they're multicellular, like the cells of plants and fungi and animals that you see here on the right. Modern science has, of course, expanded on the original cell theory, right? We've had a few discoveries since the 18th century, just a few. For example, cells contain hereditary information in the form of DNA. It's this DNA that's passed on during cell division. So, is DNA something that should categorize life? Additionally, cells carry out all of the biochemical processes necessary for life, such as energy production, metabolism, and these activities are regulated by internal structures like organelles, or organoids, which we call them in bacteria. I just want to mention that this goes back to what we said were characteristics of life, which we could change, right? We could change those characteristics. And then what is required for cells to carry out to be determined as living would change as well. All right, so thinking of that, this right now is the current, most updated understanding that we have as humans that underscores the critical role of cells in heredity, metabolism, and overall function. So hopefully in this class, we can put together some of the classical tenets and some of the modern insights that we've gotten into cell theory and form like a basis of biology, right? And we can apply that from looking at genetics all the way to looking at medicine. So before we even begin to consider what is inside a cell, it's important to think about the relative size of cells. So this diagram helps us do that. To the left on this diagram, there are things that you can see with your unaided eye, and these are macroscopic items. In this course, whether in person or online, we're going to use a microscope to view pretty much everything. There are many websites, and two of them are listed at the top right of this slide. And these websites, they kind of walk you through a relative scale. They're kind of fun. You should take a look if you can. You get a sense of the size of cells, their components, and maybe some things you might be familiar with, like yeast or DNA or carbon atoms. But these websites show you all microscopic things. There are so many different kinds of cells. And one way we create large categories of cells is to compare simple cells, called prokaryotes, to complex cells, which are called eukaryotes. Prokaryotes are things like bacteria and other fascinating organisms that are called archaea. Archaea are simple-celled organisms, and they live in extreme environments like volcanoes and hot springs and really salty lakes. We're going to consider the more complex cells called eukaryotes or eukaryotic cells. These cells have what are called organelles. And yes, organelles are to cells what organs are to your body. So we explore plant cells and we explore animal cells like those pictured here. But I want to remind you that there are other eukaryotic cells of fungi and these baffling organisms called protists that are listed on the left and right here. We discuss these organisms in the next part of this course, so that would kind of be like next semester. Another aspect of the external anatomy of cells is their surface area to volume ratio. And this is the comparison of how much covering there is to how much insides. So your cutaneous membrane or your skin is equivalent to your surface area, and it is quite large. Your insides, your squishy organs, your muscles, your bones, those are your volume. So you want this ratio to be large most of the time so that exchange with the external environment is efficient. I like to think of my cats. On cold days, they curl up, and one of them even like burrows into blankets. And their volume is fixed, right? So as she curls up, she reduces her surface area and she reduces her heat loss to the environment. But then on hot days, or when we have the wood stove going, they get like as close as they can to the stove, but they stretch out, right? And they try to increase their surface area and increase their heat loss or prevent what's really a cooked kitty.